Next up, let's go ahead and look at some active processes of membrane transport. And so just a reminder that these are the processes that require energy in the form of ATP, and they are also going to be moving particles against the concentration gradient. Okay, and so this is going to be from an area of low concentration to an area of high concentration. So let's look at some examples. The first example that we're going to be looking at for an active process is going to be the sodium-potassium pump. This is a protein embedded in the plasma membrane, and this protein is going to be moving both sodium as well as potassium, but it's going to be moving sodium and potassium against their concentration gradients. So what does that mean? Sodium is actually very highly concentrated outside of the cell. And low concentration inside the cell. So by diffusion, sodium would flow into the cell, for example, by facilitated diffusion using a protein channel. But the sodium-potassium pump will use ATP to move sodium outside the cell, hence moving it against its concentration gradient, and it moves 3 sodium ions every time. On the other hand, we also have potassium, and potassium is now highly concentrated inside the cell. Okay. And then few concentration outside the cell. And so again, by diffusion, potassium would move outside of the cell, and that would be a passive process. But in this process of an active against concentration gradient mechanism, the sodium-potassium pump is now going to move potassium into the cell, hence against its concentration gradient. And it's going to move 2 potassiums for every 3 sodiums that it moves out of the cell. And so this is one example of an active process where potassium moves against concentration gradient into the cell, and where sodium also moves against the concentration gradient, but this time it's going to move out of the cell. Now let's look at some other examples. The second example of an active process that we're going to be looking at is something that we call vesicular transport. Vesicular means from the word vesicle, and a vesicle is just a bubble-like sac that is present inside the cell, and it's going to serve to transport materials into or out of the cell. And so under the vesicular transport, we have 2 processes. Exocytosis. Exo means exit, out of the cell, cyto, cell. So exocytosis is movement of materials or substances out of the cell by the use of vesicles. And then endocytosis is another example. And it's actually the opposite of exocytosis, where now materials are moved into the cell. So let's draw a schematic to kind of illustrate what this would look like. So let's start with exocytosis. So sometimes a cell has vesicles that contain specific messengers. One example would be a chemical messenger that has to be released out of the cell. And so what happens is that these vesicles are now going to fuse with the plasma membrane, and they are now going to release the contents out of the cell. This is a process known as exocytosis, so moving the materials out of the cell against their concentration gradient, and this process requires proteins that need ATP. And so this is going to require ATP, moving materials against the concentration gradient, and this is what makes it an active process. Along the same lines, let's go ahead and look at endocytosis. So endocytosis, one example is what we call phagocytosis. And so in our body, we have cells that we call phagocytes that can actually engulf specific particles. So particles that need to be engulfed are engulfed by this phagocyte cell. And so what happened is that this is then internalized by the phagocyte. And one example would be a type of white blood cell called the neutrophil, and that's how it can, let's say, engulf or eat up bacteria. Okay? Another example would be engulfing cell debris, dust particles, foreign particles. Another example of endocytosis is what we call pinocytosis. And this is very similar to phagocytosis, is that phagocytosis, but the only difference is that phagocytosis involves solid particles, whereas pinocytosis is known as cellular drinking because it involves liquid components that are then internalized by the cell. And so these processes as well are moving content against the concentration gradient and also require ATP.